All right, welcome to this very last talk of the CCP4 study weekend. And in a way, it kind of fits to my topic because we'll talk about PDB deposition, and this is usually the kind of the last and sometimes a bit of painful step in the process. And although I hope my talk will not be as painful, as a small disclaimer, this talk will not be about like a fully developed tool, and I will not present some results with the tool. It is more about a summary about what is the current state and what are the current challenges in PDB deposition, especially for fragment screening, and kind of a small wish list how it could be in the future. And especially about what could be done with the mmCIF format. So before I go into the PDB deposition, I want to shortly introduce the F2X facility. We are part of the MX facility at BESI 2, and we are a platform that offers crystallographic fragment screening to any kind of users. We predominantly have academic users, but we are also open to industrial users. And the development of the facility started in 2012. And with that, we have already gained a lot of experience and developed well-established workflows, so we can support our users with our experience, which is one of the things that we can offer. Besides that, we have a couple of fragment libraries available, especially our F2X library, which consists of 1,000 fragments, more of 1,000 fragments, but also a subset of that. And we are developing other fragment libraries as well. And the special thing about our fragment libraries is that we have them in ready-to-use plates, where the fragments are immobilized on a crystal plate, where you can directly do the soaking. This has the huge advantage of that we can have the same concentration of fragments, despite the DMSO concentrations your crystals can tolerate. So we can always go to 100 millimolar, even if you don't have to, if the crystals don't tolerate the DMSO at all. A second advantage of this plates is that we can also ship them, so we can offer the sample preparation completely remotely. So we can, besides the libraries, ship everything you need for the sample preparation, and then you can just send us the samples back and measure them at one of our 2 beam lines. And after you have collected the data, we also offer the data analysis, which is mainly based on the Fragmex app, which is also possible to do remotely. And since last year, we also offer help to the users to deposit the structures into the PDB. Before I start talking about depositing fragment screening data, I want to introduce you a little bit to the mmCIF file format in general. It will not cover everything, but I hope everything to understand the later parts of the talk. The mmCIF file format is short for Macromolecular Crystallographic Information System, and it's the current file system to deposit structures into the PDB, and it replaces the old legacy PDB file format. Some of you are still quite familiar with, I guess. The first version was released in 1997. And it became the standard archive format in 2014. And since 2019, it's mandatory to submit the structures in mmCIF format to the PDB. And an initial, one of the aims for the mmCIF format was to overcome some limitations of the PDB format. For example, in the old format, it was not possible to deposit larger structures, like from complexes. There was a limitation on it, and in the new mmCIF file format, there's no limitation. And there are also more options to store metadata in there, as well as in a more structured way. And it is better machine readable, although I would say it has compromised a little bit on the human readability, I think. So here you can see an example of a beginning of an mmCIF file, so it's much longer, but you already see it's a lot of text, and I want to walk you through the general architecture organization of such as an mmCIF file. The top organization level is called a data block. And usually a data block contains like information for one structure or for one, like an electron density map. And it always starts with data underscore, and then it can be followed by the name for the data block. This will become a little important a little bit later. The next organization level are the categories, which contain the related information like the cell parameters or the position authors or whatever, so things that can be grouped together. And they are always separated by the hashtag or a space, or I mean an empty line. And within the categories, you find item value pairs, and they contain individual information. And here's an example of one of these item value pairs. You have, I picked from the cell constants, the cell axis, the A, and it has a value. And the item name is also a composition of the category name and an attribute name separated by a dot. And this is just to clear some language. And then I want to give you an example of one of the categories. So you see here again the cell parameters, and there are in general two ways on how the categories are represented in an mmCIF file. One is as a key value pair, which you can see here. And another one I will show in the next slide. It's about loop representation. And I want to encourage you, whenever you look into such a CIF file and see these categories, I want to encourage you to think about like tables. So you can, in principle, convert each of these categories into a table, which I have done here for the cell parameters. Yeah, and when we think about the tables, the category name is the same as would be the same as the table name, and each of the attribute will be converted to one column, and the volumes are then the entries of the table. And here's the second example with a loop representation. So this is needed if you have more rows. If you have for one item more than one value, it's written, it starts with a loop underscore, and then you just get the column names basically, and then the values in a tabular format. And this I converted to a table as well. So I guess this is a little bit better readable. Yep. So now with all of this background information, we can go into what is the current state of deposition of fragment screening data into the PDB. And I'm gonna speak about what we are doing at the F2X facility. And at the moment, the first step is to look at our data sets and to decide which data sets we want to submit to the PDB. So there's the possibility to deposit only the hit data sets, or also the ones that were used for the ground state calculation for Panda, or just all of them. This at the moment we leave to this decision we leave to the users, whatever they want to do, but we offer that we can deposit hits as well as ground state models. Then we have to prepare the mmCIF files for the structures and the structure factors. And here comes the pipeline in that I have developed. It's called FTX to PDB. And I started it last year, so it's still quite in development, but we have successfully deposited some data with this workflow. At the moment, it's a command line tool, but I would like to put a GUI on it so that people can use it more conveniently. And at the moment, it is based on PDB extract and GAMI to convert the files and modify them. And right now, it's a little bit tricky to use it still because there are a lot of changes happening. And so right now, it requires someone from the FTX team to help to use this tool and to deposit the data. And then when we have prepared all of the mmCIF files, we deposit the data via the group deposition tool, which is possible via our FTX access to the group deposition tool. So coming back to the mmCIF files, what currently is available to support these group depositions to deposit also fragment screening data is shown here as an example. We have here the category about the group deposition, and right now you can specify, you will get a group ID and you can give it a title and a description, and you can assign a type. So if it's a hit state or it's a hit, or if it's a ground state model. But this is in fact all what we have right now to deposit something into the PDB information about fragment screening data. So in reality, it still looks more like this. There's many open challenges, and I grouped them roughly into 3 categories. The first one is the development of better tools, which I think we're currently doing, and it still needs just some more time. I hope we can make our pipeline a bit more robust and also give it a better user experience with the graphical user interface. Then there are a lot of open questions and sometimes also some disagreements in the community about how to publish fragment screening data. Especially we have missing guidelines and standards so that all of the facilities come to the same conclusion, like what do we want to publish and what not, and in which way this can be standardized. And also there have also been people who have raised concerns about depositing fragment screening data into the PDB, and I'll show a little bit about this as well. But then the third big group is the limited possibilities to add metadata from fragment screening. So fragment screening specific metadata into the mmCIF file. As I showed, only this group deposition information can go there. And for example, right now we just put the fragment name into the title and things like that. And one important thing I want to mention right now is like that the group deposition does not equal fragment screening data. I think that gets conflated quite a lot, but there are also group depositions which contain not fragment screening data, and also fragment screening data is getting deposited outside of group depositions. One of the major criticisms is about depositing fragment screening data into the PDB is that it could mislead users. Right now, some people are depositing manually refined structures of their hits, but we also publish just the auto-refined structures. So there's a mixture of this. And as I said before, there's also a mixture of group depositions and single depositions. So, and it has become a bit of a problem that these group depositions or fragment screening data has gotten the assignment of like having lower quality, which I think is not exactly true, because as I said, some people do refine their structures, and I think not all of them of the auto-refined structures are as bad as it was sometimes said. Yes, there are some structures which have problems, and we have learned from that, and we need to implement filters and checks in our pipelines that we don't publish this kind of data as well. But what the concerns are is really that if there's data of lower quality, whatever that means, in the PDB, that could mislead users, especially inexperienced users who don't know that this is other type of data. And it also has been said that it might be a problem for AI development if there are lower quality structures. And I think it is overgeneralized because, as I said, they have also good structures in there. And the claim was that there should be a separate database for fragment screening data, or at least a clear label that this is fragment screening data. And the second point is like it was said there's a missing evidence in the electron densities, because it was only looked at the normal F2 FO minus FC maps, and not at the panda maps. Some fragments are binding only at the lower occupancy and can only be identified via panda. And if the panda maps are not available or not looked at, then you could, of course, get the impression that we modeled just some weird stuff in there that is not there. But here you see an example of one fragment where there is clear evidence in the panda map. And this one is an example of how the mmCIF file can already be used to solve this problem. Because as I said before, there's the possibility there are these data blocks, and there's a possibility to have several data blocks within an mmCIF file. You can go to the next slide. And so you can put more than one MTZ file, converted MTZ file into the mmCIF file. So what we are doing right now is we have everything in there. We have the process data, we have the refined maps, and the panda maps, if the hit was identified via panda. And we deposit this as the structure factor file into the PDB. The only problem right now is that you have to separate these three data blocks before you want to visualize them. But in principle, everything is there and it gets deposited. What is not there, what I mentioned before, is the possibility to deposit other kind of metadata. And for this, we propose that will be additional mmCIF categories, especially for fragment screening metadata. And with this, there comes a lot of possibilities. This would, for example, provide the possibility to just build in a flag, like, is this fragment screening data or not? So you can simply filter it. You can then, whatever the community decides, if we want to have them labeled in a PDB or not, all of this can be implemented, and it can also be a filter step, could also be used for AI development. So I think this could be really beneficial. And we could even go beyond the idea of having a flag like, is it fragment screening data or not? We could also implement something like, is it auto-refined or manually refined data? So we could even make the separation or this filter, and also like things like, is it a hit or is it ligand bound? Could be implemented like this as well. And now I would just go briefly through the other ideas we have. I don't go through each and every information that we would like to have in there, just to exemplify it. And I categorized it now like on several levels. The first level is something is on information on a whole project of fragment-based drug discovery. So even more than a fragment screening campaign. So sometimes for some projects you run several campaigns, or you do follow-up screens. They could all be combined under one FBDD project, and this could get an ID, for example, and then you could add some more. Here you could, and this could provide easier access to find, or it can be easier to find related data. The next level is on is the level of the fragment screening campaign. So here you could we could possibly put everything in that is kind of the same for each data set in the fragment screening campaign. For example, information about which fragment library was used, at which screening facility the data was collected, if it was collected in the screening facility, and like experimental procedures, how the fragments were soaked and things like that. The next level is on the dataset level. Here you could add information about the compound. As I said before, right now we're just putting the name of the compound into the title. This is very difficult to extract at some point. You could even say, like, if it was, especially for follow-ups, it was specially synthesized or purchased from somewhere, and like where in the library it can be found, things like that. And also here, this filter could be like if a ligand is bound or not, or if it is a hit or not, could be implemented in here. The fourth level is on the binding site level. In some cases, you have several binding sites of a fragment, or you have several monomers in the asymmetric unit. So you want to have also the level of each binding event that you can assign. Like, for example, is it a hit, or is it an interesting hit? Is it a binding? Is it the active site, or is it in a crystal contact? Things like that. Or if it's interesting site for the drug design. And the fifth one is about follow-up strategies. So I think here's also a lot of potential for AI development. If we could capture this kind of information, for example, if a follow-up, we could link it to we could have a reference to the initial hit. Also, we could also say like which strategy was used, and you could assign like if it was a good data follow-up strategy or not. And the last one is about Panda or other multi-data set analysis tools. So here we could implement something like if the hit could only be identified by Panda, or which data sets were used for the ground state model. And in principle, there's a lot of possibilities to extract the information from Panda output, from Panda analyze and Panda inspect. So as conclusions, there are still a lot of open questions that have to be answered within the community. There will be a workshop about this in March, so I hope we can get to some conclusions at that point or in the follow-up of that. For example, like if the fragment screening data should be deposited into the PDB or a separate database, and if it's deposited into the PDB, shall they be treated differently or labeled or whatever? Which data sets should be published, PIPs only or everything? What could be the standards on the metadata, which could be implemented in the mmCIF file? And then, as I said, it would be great to have these categories in the mmCIF file to allow us to publish our metadata as well. And also then right now the access to the group deposition is limited. You need credentials to log in, but it would be great if this could be open to everyone and that they don't have to rely on the facilities to do the deposition. So overall, there's a high need to publish fragment screening data in a standardized way, because I guess there are a lot of unpublished campaigns lying around. I have several of them as well. And this could also be a great foundation for AI development. And with that, I would like to thank everyone who is involved in this project and everything about the facility. First of all, the F2X team, especially Laila, who kind of initiated this journey by last year coming to me and asking me like, hey, can you help me with depositing my structures? And I said, yes, of course. And now I'm here. So then the students and the users, they are the greatest source of new ideas on how to improve the user experience, and everyone else of the MX team at BESI too, as well as the funding and all of the collaborations. And finally, thank you for listening, and yeah.